Good morning, everyone. And today the topic of my presentation is polymerase chain reactions, principle and practices. This presentation sessions covers about the background of PCR techniques, types and their application. Take a look at the introductions about the PCR. PCR is a laboratory versions of DNA replications in cells. It means it is an in vitro techniques for amplifications of DNA. In 1985, Carrie B. Mills invented PCR techniques, and he also awarded Nobel Prize in Chemistry in 1993 for the same work. Terms comes polymerase. So why it is called the polymerase? Because the only enzymes which is used in this reaction is DNA polymerase, and the chain because the products of first reactions become substrate of the following one and so on. So basically PCR is a cyclic process and it consists of a series of 30 to 35 cycles. The purpose behind this technique is to make a huge numbers of copies of your targeted gene or interest of gene. In simple word, we can also say that it is an automated versions of DNA replication, or we can say it's like a Xerox machine for gene copying. A major component of polymerase chain reactions are DNA template, primers, DNTPs, buffer solutions, divalent cations, and sterile deionized water. We can see we can see from the diagram the PCR components, and this is the PCR tubes in which PCR reactions take place in a small polypropylene tubes, and this is a thermal cycles in which a reaction is automated by using a thermal. The first component of PCR is DNA template. So DNA template, it is a that particular DNA sequence which you want to be copied, or we can say, or we can, or we can say it is the DNA molecule that contains DNA region to be amplified. The segment that we are concerned, which is the target sequence. The next one is the primer, and primer are the short oligonucleotides of DNA, and it provides a starting point for DNA synthesis, which you want to amplify. So they can be the random sequence. They can be random sequence if the project goal is for general genomic studies. However, uh, if the purpose is to amplify a certain sections of DNA in the genome, uh, such as known gene, then primers of the specific sequence must be used. So basically, the two types of primers are used, the reverse and the forward primers. And these two types of primers are basically used in the commercial PCR reactions, which is designed to the flank of the target region for amplification. And primer should generally have the following properties like 18 to 28 bases of length, 40 to 60% of GC content, have a balanced distributions of GC and AT rich domains, and 50 to 60% of melting temperatures. And primers pair should not have complementary regions. After the next PCR component is the DNA polymerase, and DNA polymerase, which is the type of enzymes that um, synthesizes the new strands of DNA complementary to the target sequence. The first and most commonly used of this enzyme is TOC DNA polymerase, and it is a highly thermostable recombinant DNA polymerase. It is named after Thermus aquaticus, the heat-tolerant bacterium from which it isolates itself. Whereas another DNA polymerase name called PFU is also widely used because of its higher fidelity when copying the DNA. The next component is DNTPs. The function of DNTP in PCR is to expand the growing DNA strand with the help of the TOC DNA polymerase. It binds with the complementary DNA strands by hydrogen bonds. Basically, four DNTPs are used in PCRs. They are like DATP, it means deoxyadenosine triphosphate, and another one is DGTP, which is deoxyguanosine triphosphates, and deoxycytidine triphosphates indicates DCTP, and DTTP, it indicates deoxythymidine triphosphate. And after that, finally, a reactions buffer is used. Why we need to use a buffer? Because a buffer is used to provide a stable pH. It may also contain magnesium chloride. If not, then MgCl2 must be added separately, because the divalent cations like Mg2 plus, it's play a vital role in PCR reaction, and it act as a cofactors for TOC DNA polymerases, and thereby influencing its enzymatic activity. Let us come to steps involved in PCR. Conventional PCR involves 25 to 30 repetitive cycles, with each cycles comprising three sequential steps, and these are the denaturations, annealing, and extension steps. This is the pictorial diagram of PCR's procedures, which clearly shows the first steps is denaturations. In this step, the reaction mixtures, here the reaction mixtures is heated to 93 to 98 degrees Celsius for a short time of periods, about 15 to 30 seconds, to denature the targeted DNA into a single strand. Here we can see, and that can act as a template for DNA synthesis. The next step is primer annealing. In this, the mixtures is rapidly cooled to a defined temperatures, which allows the two primer, here the two primers, to bind to a sequence on each of the two strands which flanking the targeted DNA. And this annealing temperature is calculated very carefully to ensure that the primers bind only to the desired sequence. And the temperature is usually 50 to 55 degrees Celsius. Here the third steps is an extension steps, and in this steps the temperature is increased to 70 to 75 degrees Celsius, which is the optimum for DNA polymerase activity. And DNA polymerase activity allow to hybridize the primers to be extended. Or we can say that the purpose of this step is to allow the re-annealing of the PCR products into double standard DNA. And the all these three steps are repeated for a third cycle, and so on for a set of additional cycle. By the third cycle, some of the PCR products represent DNA sequence only between the two primer site, and the sequence doesn't extend beyond these sites. So we can see from the here we can see from the diagram, as more and more reaction cycles are carried out, the double standard DNA are synthesized more in number, and after 20th cycle, the original DNA has been amplified a millions of copies and rise to a billions of copies after the 30th cycles. Here we can see. And after the PCR once has been completed, methods called electrophoresis can be used to check the quality and size of the PCR products or fragments produced. This is the variants of the PCRs. Basically, PCR is a nucleic acid amplification techniques. So when we talk about the DNA amplifications, it divides into four parts, and they are the one-step methods, two steps, multiplex, and modified PCR. In one step, it contains conventional PCR, real-time PCR, mini PCRs. In two steps method, It contains nested PCR, assembly, methylation mediated ligations, and in modified PCRs, it contains hot start PCR, touchdown, in situ, allele specific, inverse, and solid phase. And when we talk about the RNA amplification, we will use the reverse transcriptase PCR. In this short sessions, it quite impossible to explain all these techniques. So I'm trying to focus over the major topics which is used in research areas for today era. Let's start with the Rt-Pcr. It stands for reverse transcriptase PCR. Um, it is often confused with qPCRs. However, they are the separate techniques, separate and distinct techniques. So they are the separate and distinct techniques. So it is used to qualitatively detect gene expression through creation of complementary DNA and transcript from RNA. And whereas qPCR is used to quantitatively measure the amplifications of DNA using fluorescent probes or dye-based probes also. So we can say the Rt-Pcr is a technique which is used in genetic studies that allow the detectations and quantifications of mRNA. And in Rt-Pcr, the RNA template is first converted into cDNA using a reverse transcriptase enzymes. And the cDNA is then used as a template for exponential amplifications using PCR. We can see the procedures of Rt-Pcr. Basically, the two main methods, the first strand reactions and the second strand reactions. In first strand reactions, the mRNA strand is first reverse transcribed into a single standard DNA, into single standard complementary DNA temp, into single standard cDNA using DNTPs and RNA-dependent DNA polymerase, which is reverse transcriptase, through the process of reverse transcription. For second strand reactions, after the cDNA is after the cDNA is generated, the standard PCR is initiated after 30 cycles, or the million of copies of the sequence of interest are generated. And after that, the original RNA template is degraded by RNAs, but leaving the pure complementary DNA or cDNA. Discuss about the requirements of Rt-Pcr, and from the diagram we can see how they proceed. In Rt-Pcr, depending on the requirement, primer for first cDNA synthesis can be of the two types. The first one is oligo DT, and the next one is the reverse primers. From the diagram, we can see here the oligo DT, which is used as primers, which binds to poly A tail of mRNA. And second one is the reverse primers, and they are specifically designed which can hybridize to a particular target genes or defined mRNA family. And the most commonly used and commercially available reverse transcriptase enzymes are AMB and MO-virus or MORT. When we talk about the quantifications of mRNA using Rt-Pcr, it can be achieved as either one steps or two steps reactions. This is the diagram of one steps and the two steps reaction. The difference between the two approaches lies in the number of tubes which used when performing the procedures. So basically in one steps reactions, it requires the reverse transcriptase reactions and PCR amplifications which perform in separate tubes. However, the starting RNA template, the starting RNA template are prone to degradation in one steps approaches, and use of this approach is not recommended when repeated assays from the same sample is required. So in two steps reactions, the reverse transcriptase reactions and PCR separations performed in separate tubes, and in one steps, the entire reactions from cDNA synthesis to PCR amplifications occurs in single tube. So both these two different steps are depicted here, and we can see here this is the one steps and this is the two steps reactions. Common applications of Rt-Pcrs, it includes in the various way. It is used in research laboratories to study the gene expression. For example, in experiment to distinguish exons from introns. And it can also be used clinically to diagnose genetic diseases and monitor drug therapy. It's also useful in generating the large cDNA libraries from the small amount of mRNA. And it also used in cancer detections to help improve prognosis and monitor response to therapy. Let's say with an examples, we all know that the circulating tumor cells produces unique mRNA transcripts depending on the type of cancer. So the goal is to determine uh, that which mRNA transcript serves as the best biomarker for a particular cancer cell type, and then analyze its expressions levels that we use Rt-Pcr. And Rt-Pcr is commonly used in studying the genomes of viruses whose genomes are composed of RNA, such as influenza virus, retrovirus like HIV, and SARS-CoV-2 also. The technique is a real-time PCR. It is a variant of PCR. It is a variant of PCR technology. It is also known as kinetic PCR, qPCR. Basically, it is a quantitative PCR methods that enables for the identifications of PCR products as they in real time throughout the amplification process. It monitors the fluorescence emitted during the reactions uh, as an indicators of amplicon productions at each PCR cycle. So it's commonly used to determine whether a DNA sequence is present in a sample and the number of its copies in the sample. Principle of real-time PCR depend upon the use of fluorescence dye. The two types of chemistries are available for the real-time quantitative PCR. The first one is the DNA binding dye, which is also called the dye-based method, and the second one is the sequence-specific probes, or we can say the probe-based methods. In dye-based method, the dye has its own fluorescence. Once the dye binds to the double standard DNA, here we can see from the diagram, this is the double standard DNA, and this the green color indicates the dye. So once the dye binds to the double standard DNA, the fluorescence emitted by the dye and increases 100 to 1000 folds than the original signals. However, the original dye fluorescence is taken as a baseline, as a reference for the detectations. And this dye-based method is rapid, quick, reliable, and very cost effective. And also the chances of error in this method is less, and the reaction setup is very simple and easy to use. From this pictorial diagram, we can see how the dye binds with the DNA. When fluorescent dye, see, when fluorescent dye binds with the double standard DNA, increases the fluorescence. For examples, we use SYVR green dye, which is most widely used in double standard DNA specific dye reported for the real-time PCR. And it binds to the minor group of the double DNA helix in the solution. The unbound dye exhibits very little fluorescence. Apart from SYVR green dye, we use ethidium bromide can also be used for the detection. But due to its carcinogenic natures, it used restrictive. Another methods is the probe-based methods. Uh, so specific detections of the real-time PCR is done with some oligonucleotide probes labeled with both a reporter fluorescent dye and a quencher dye. And these are the Tocman probe, hybridization probe, Scorpion probe, and molecular beacons. This is the comparisons of SYBR green and the talk band method, or we can say the dye-based and the probe-based method. From this diagram, we can see the top probe-based method. Basically, the Talkman probe is a short DNA sequence with a high energy dye called the reporter dye, which tagged at the 5' end. The 5' and this is the 3', 5', uh, 5' end, and the quencher dye, which is tagged at the 3' end. When these probe is intact and excited by a light source, the reported dye emissions is suppressed. After that, as a result, due to the when these probes is intact and excited by the light source, the reported dye emission is suppressed by the quencher dye as a result of close proximity of the dye. And this probe is used for the DNA quantifications, and it also used for mutation detection. Some other probes are also used. I already told hybridization scorpions and the molecular beacons. So basically the uh, molecular beacons is a hairpin shaped molecules and having an internally quenched fluorophores whose fluorescence is restored when they connect to the targeted nucleic acid sequence. And this is the novel and non-radioactive method for detecting specific sequences of nucleic acid. This is the diagram of dye-based method, which I already discussed in previous slide and how they work. The graphical representations and the models of the real-time PCR plot. Amplifications curve present into three different phases. First one is the initiation phase, the second one is the exponential, and the third one is the plateau phase. Here we can see from the graph the first line is called the initiation phase. It occurs during the first PCR cycles where the emitted fluorescence cannot be distinguished from the baseline. And the baseline is the place where the fluorescent signal shows no or little changes. After that, during the exponential phases or the log phase, increases in fluorescence before the plateau phase reached, and it is also called the threshold cycles or the CT, and it indicates that the cycle number where the reactions fluorescence crosses the threshold, and this reflects the point in reactions when the sufficient amplicons have been generated to give significant fluorescent signals over the baselines. In the last phase, the reagents are exhausted and no increase in fluorescence, and it is the last phase of the and it is the phase where observed the and it is the phase where the standard curve observed. And this is the point considered as standard curve. So only in the exponential phase, quantification is possible. Applications of real-time PCR, it is used in the field of clinical microbiology, oncology, and the gene therapy. And it is clear that it has become the standard methods for disease detections. It is also used by microbiologists in the field of food safety, food spoilage, and fermentations, and for the microbial risk assessment of water quality. In the field of research, it provides the quantitative measurement of gene transcriptions. It has been shown to extremely useful for the quantifications of viruses, bacteria, protozoas, and many parasites. The next technique is the nested PCR. It is a modifications of PCRs which is designed to improve the sensitivity and amplifications of DNA. And the advantage of using this PCR techniques is to low probability of the non-specific amplifications, because in the conventional PCR, the RNA primer can go and bind to different regions, or we can say unwanted regions of the DNA, and uh, we block the unwanted binding region of this primer by utilizing the nested PCRs. And so in nested PCR, two sets of primers are utilized. In the first reaction, the first pairs of primers, which is known as outer pair, is used to generate DNA products, in which amplify a fragment which is similar to the standard PCR. And the second pair of primers, which is the nested PCRs, which bind inside the first PCR product's fragments and to allow the amplifications of a second PCR product, which is shorter than the first one. This pictorial diagram shows the overview of process. So this is the DNA, the first PCR run and denaturation process is done. Then bind the primer, and this is the first set of primers which can bind both the sides. This one, green color. Side of diagram, we can see the some of the primers which bound to unwanted regions. And from the right hand side, this is the primer which bound to the actual target sequence. After that, both will start amplifications. After that, both will start amplification. So one of the segment produces this product which is totally unwanted. So this type of problem basically creates in normal type of PCR. And so after all PCR process done, we got lots of unwanted products in PCR tubes and very few wanted target sequences. And it will be very tedious work to separate the target sequence, and it will take extra time. So from beginning, we will use two set of primers so that will bind to exact the target sequence. And from that and from the diagram, we can see it bind to inner side of the target sequence, and this red color indicates. And after binding, it will only amplify the segment of target sequences, and we get this part, the uncontaminated and the final products. It is very beneficial in studies such as the phylogenetic analysis and the genetic polymorphism. The main advantage of the present methods is that it gives 100% accuracy, specificity, and sensitivity. It is also useful in the amplification of genes with the low abundance, and used from the long time in pathology labs from microorganism detections for the detections of rickets and many organisms in tissues and bloods. It also used for the detections of herpes virus in the CSF and tuberculosis in the sputum samples. And it also used for the study of viral infections and carcinoma. The next techniques is the multiplex PCR. So multiplex PCR is a widespread molecular biology techniques for amplifications of multiple targets in a single PCR experiment. So in this technique, sequence can be amplified by using multiple primers in a reactions mixtures. Annealing temperatures for each of the primer set must be optimized to work uh, correctly within a single reaction. And amplicon size, that means their base pair length, should be different enough to form distinct bands when visualized by gel electrophoresis. And this is the diagram of the multiplex PCR. This is the different types of targeted which we want to amplify. The two types of multiplex primers, basically the two types of multiplex PCRs used. The first one is the single template PCR reaction, and the next one is the multiple template PCR reaction. In single template PCR reaction, this technique uses a single template which can be genomic DNA along with several pairs of forward and reverse primers to amplify specific region within a template. And another one is the multiple template PCR reaction. This technique uses multiple templates, several primers set in the same reactions tube. Presence of multiple primers, it may lead to cross hybridizations with each other and the possibilities of the mispriming with other templates. So when we design a primer, some parameters should follow. The design of a specific primer set is essential for a successful multiplex reactions. So the important primer design considerations points described below here. The first one is the primer length. Multiplex PCR assay involves designing of large numbers of primers. Hence, it is required that the designed primer should be of appropriate length. Usually, primer of short length in the range of uh, 8 to 22 bases are used, and the melting temperatures between 55 to 60 are used for sequence with high GC content and primer with higher temperatures. Specificity, it is important to consider the specificity of the design primers to the target sequence while preparing a multiple assay, especially since completion exists when multiple target sequences are in a single reaction tube, and avoid the primer dimer formations. And the design primer should be checked for formations of primer dimers with all the primers present in the reactions mixtures, and because the dimerizations lead to the unspecific amplifications. This techniques has been used to screen for individuals of or symptoms associated viruses, and examine association of virus infections with disease. And in addition, this techniques has been shown to be a very powerful and cost effective tool for typing and subtyping viruses strains in different epidemic in different studies. It has been found very useful in pathogen identifications, high throughput SNPs, genotyping, mutational analysis, uh, gene, de gene detections analysis, template quantifications, linkage analysis, and RNA detections and forensic studies. Now let me give you summary of what I have been talking about over the last hours. So the goal of PCR reaction is commonly to replicate only a portion of the genome of interest, and it is used for the wide range of applications in science, industry, medicines, agriculture. It is also used in health and medicines. It is used to advance our understanding of cancers and human genetic diseases such as Parkinson's, cystic fibrosis. It is very important to the genetic identifications of fungal, bacterial, and viral diseases. The future of PCRs appears promising, and it also helps to determine the maternity, paternity, and uh, other blood relationship. And it is used by forensic scientists to identify individuals to determine, and it also used to determine the plant species, and it is used in agriculture field, pests and diseases.